Hello students, now we will start with new topic, that is Hardy-Weinberg equilibrium. It is also called as Hardy-Weinberg principle or the mathematical expression regarding the frequencies of alleles of the genes in an algebraic expressions. So a Hardy-Weinberg principle, it is also called as Hardy-Weinberg equilibrium. It is also called as frequencies of allele forms in population genetics. The numericals or the values here are expressed in algebraic expressions or mathematical expressions. This Hardy-Weinberg equilibrium or Hardy-Weinberg principle was given in 1908 year. It is given in 1908. So, if we see once again the definition, Hardy-Weinberg equilibrium, it states that the frequency of alleles of a gene on the loci of a chromosome that remains the same. It means that same, we can also use the term like constant. For suppose there was some number of parents, F1 generation, F2 generation, F3 generation, etc. Suppose think that they are having some constant number of genes. We can take here number according to the type of species. For suppose, here we have to use a term like gene pool. Here the term gene pool is considered. What is meant by gene pool? Gene pool is the sum total amount of all the alleles or the genes in that particular population. The sum total of all genes in a population. Means here we also learn about genetic equilibrium that is maintained at constant level in all the generations. So we will write here genetic equilibrium is also maintained. It means that if the parents are having some particular number of genes or alleles, the same number will be the number will be passed to F1 generation. For suppose, I am not taking here a population, just one simple example to explain this one as same or constant. Parents are having, human beings are having 46 number of chromosomes, the gametes are getting half, but still we say parents are having 46 number of chromosomes. So 23 plus 23, 46. From here, again F1 generation, each and every offspring will get separately 46 number. When again they are getting reproduced, again the F2 generation will be getting 46 number only. Again the F3 also gets 43 only. So here I told just an example regarding the individual. The same is applied for the population. Means the total gene pool or the frequency or the sum total of the alleles in a gene pool are same in the parent. The same numerical value is seen in F1. The same numerical value is seen in F2. But it varies from species to species. So we can represent this one this Hardy-Weinberg principle as sum total of allele frequencies is equal to 1. In the same way, individual frequencies of alleles are expressed as P and Q. P and Q. If you take the parents, so let's take this one as a loci, and here is dominant alleles pair, that is capital A, here I am writing. We can also take it as P, capital P and capital P also. This is crossed with homologous pair of chromosome, which is having the allele pair as small q and small alphabet q. We can take this both as parents. We know that when such type of homologous chromosomes are crossed, here we have used a term like during sexual reproduction. So in the pachytene there will be crossing over and there will be exchange or gene recombination at pachytene stage. In F1 generation, since here the alleles are homologous dominant, here are homologous recessive. In F1 generation, we get heterozygous dominant, which is phenotypically expressing the dominant feature. In F1 generation, we are getting the heterozygous type of allele pairs, which are crossed from homozygous parents. So at this stage, we can represent like in an population. So in an population, there will be dominant alleles, there will be recessive alleles. There will be random mating among the population. What is meant by random mating? According to Hardy-Weinberg equilibrium, the population sample that we are taking for calculation should have random mating. Means here the dominant allele containing organism is there. It should be able to mate with the another recessive one containing organism, or it can mate with another dominant containing organism, allele containing organism. This type of mating is called as random mating. There is another word, that is non-random mating. For suppose there is an recessive allele containing organism, it will pair only with recessive containing organism. Such type of mating is not called as random mating. It is called as non-random mating type of organisms. This random and non-random mating type of words are meant when the organism is selecting the different or same type of phenotype character containing organisms. So here homozygous dominant is pairing with homozygous recessive allele pair of chromosome. But here in F1 generation, this population we can represent according to Mendel's monohybrid cross. So we have got this one after crossing with homozygous parents like this. Here if you take out percentages, it will be 25%, it will be 25%, it will be 25%, it will be 25%. Means overall the F1 generation is representing 100% of gene pool or allele frequencies. Then what about parent? Here we are taking only two homozygous it is equal to 50%, it is equal to 50%. Here also coming to parents, it is 50% and it is 50%. So parents also showed same type of allele frequencies. Population, if the number of organisms in a population has to be increased, there should be increase in the number of generations. So F1 organisms will self-mate. This possibility is also there in the F1 generation. So we can represent this one in an Punnett square. We are getting F2 generation like this when F1 generation is self-crossed. If we take out this one separately, the parents are of 50%, parents are of 50%. So here we have taken two different parents which are heterozygous, and this is showing 50% of allele frequency, this is showing 50% of allele frequency, same type. Coming to the offsprings, if we consider this is 25%, 25% of allele frequency, 25% of allele frequency, and 25% of allele frequency. All of them are constituting totally 100%, it is okay. But if we divide these 25%, pure homozygous dominant alleles in F2 generation are of, these are considered as the offsprings, 25%. Then what about heterozygous? These are 25 plus 25, that is 50% of allele frequency. Next, homozygous recessive allele paired organisms. These are of 25%. If we add, we are getting 100% in all. Means the sum total is also same as like that of parent. Parental generation, F1 generation, F2 generation. In every generation we are seeing complete 100% of the allele frequencies. In Hardy-Weinberg equilibrium, we study the frequencies. The law of frequencies are genetic frequencies. In this Hardy-Weinberg equilibrium, the frequencies are genetic frequencies. We know that whenever we are writing about genotype, it means alleles are present in the paired forms. We see here alleles are always in paired forms. The same one, the same percentages here, we will see in terms of frequencies now. If we take out 25% of homozygous dominant alleles, it means that 25 by 100, it means 25 1 4, 1 by 4, that is equal to 0.25. This is considered as the frequencies of dominant alleles. Now, another one. Capital A, small a, that is heterozygous type of alleles. Here we have got 50%. If you represent this one in frequencies form, 5 1s, 5 2s, that is equal to 1 by 2, that is 0.5, sorry, what percentage? 0.5 is the frequency. Coming to homozygous recessive one, we have seen it is 25%. If we write in terms of frequencies, then it will be 25 by 100, that is 1 by 4. Again it is 0.25. Means this is equal to 0.25. This is equal to 0.5. This is equal to 0.25. If we add all those in the gene pool, we are telling 0.25 and 0.5 and 0.25. If you add some 1, we are getting 1 as answer. Means if we add all those also, the sum total of gene pool is the same, is constant for that particular species in a population. Okay. These are two number of alleles. So we can write here as A square plus heterozygous are 2 in number. So 2 capital A small a. Homozygous are 2 number of alleles in the means these are nothing but gene forms. These also can be written as, so we can write the sum total of all the alleles or their frequencies. Frequencies, I said you that individually they are expressed as capital alphabet and small alphabet. So it is written as 1. But Hardy-Weinberg complex is mostly explained by taking the alphabets like capital P, next Q, that is equal to 1. But here, if we write here as this is P and Q, this is P and Q, and this is Q and Q. We can write P square plus 2PQ plus Q square, that is equal to 1. 1 is considered as constant for that species. There is another representation of expression of the frequencies of alleles. So Hardy-Weinberg equilibrium also expressed as one dominant allele plus recessive allele, means individual frequencies of the alleles is also constant. So we have two different equations here for calculation, that is P square plus 2PQ plus Q square is equal to 1, that is constant. P plus Q is also equal to constant. So if we represent the values in these both equations, if we take one P plus Q, if we represent here, so it is of 50%. So first we will take this equation and we will apply. So we are writing here P square plus 2PQ plus Q square, that is equal to 1. I am writing this value is 0.25 according to that Punnett square, plus we don't know about this one separately, plus Q square is again 0.25, that is equal to 1. Now we have to find out this heterozygous 2PQ as P square is equal to 0.25. If we represent this one like this, then answer is 0.5, 0.5. So we have got P. Then apply the value of P in the above equation. Since P plus Q is equal to 1, we got here P as 0.5 plus Q is equal to 1. So Q is equal to 1 minus 0.5. Again it will be 0.5. Now we have got the values of P and Q separately. So we will fill this one as 0.25 plus 2 into 0.5 into 0.5 plus here the value 0.25. If we add all these, then we will get answer 1 only. If we do little bit calculation, like we are adding this 0.25 plus 2 into 0.05 into this one. 0.05 into 0.05, 0.25 into 2. Then answer will be 0.5. If we calculate also 0.25 into 2, 2 5s are 10, 2 2s are 4, 5, 2 zeros are 0. 0.5 only we got. Again we are adding the homozygous recessive one, 2, 5. Then also answer is 0, 0 and 1. We got 1 is equal to 1. So in this way we can calculate Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium is having few conditions to explain all these calculations. What are those conditions? If we take a population as a sample, population should be large in a number, one condition. Second thing is, mutations should not be seen in that population, because if mutations are there, then there will be change in the gene pool. Next one, genetic drift should not be there. Genetic drift in the sense, from a population, if few number of population has been exited out and those few number of organisms have developed a new population at another place, that condition is called as genetic drift. So, Hardy-Weinberg equilibrium is applied or can be explained only under few conditions. We can write here happens also. Happens only under few conditions. What are those? First thing is, population should be large. Second thing, there should be no mutations. Third condition, there should be no non-random mating. Means the Hardy-Weinberg population should express random mating. Next, there should be no genetic drift. Next, fifth condition, there should be no migration. So population should be large in number. For suppose, if the population is already small, if some of the organisms have been dead, then the frequencies will be differentiated. There should not be mutations. If mutations are there, there will be change in the gene pool. Next, no non-random mating. All organisms should be able to express random mating only. Next, there should be no genetic drift. Organisms should not move out of the population to develop new type of population at some other habitat. There should be no migration. Means there should be no immigration, no emigration. No immigration and there should be no emigration. So this is about Hardy-Weinberg equilibrium. We will see some of the points that are mentioned in NCERT textbook. First one is, speciation means it is a process of how organisms that migrated to new habitat are separated and developed. their own unique characters. Here, this term is different from that of genetic drift. In genetic drift, we are dealing about the total gene pool in the newly separated population. But here we are dealing with the process of how the separated population has developed unique characters and survived in the new environment. We can take here some of the examples like cichlid fishes, means a small aquarium like colorful fishes. We can take squirrels, and we can take some Darwin finches also can be considered here. Darwin finches. Some of the plant species like Raphanus brassica, which are hybrid varieties that are having tetraploidy, obtained from two diploid organisms and obtained a new character, that is tetraploidy. Another one is Triticum durum. This is having hexaploid, that is 6N condition. So these are some of the examples. We can also consider some of the amphibians that have been developed into that have developed and adopted to the new environments. So this is just speciation, means just a process. Then what is meant by adaptive radiation? Basing on the single character or the single point of ancestral characters, organisms have developed many of the characteristic features. So basing on one single ancestral character, new phenotypes and new genotypes have been evolved. Here also we can take example like Darwin finches. Then another term, that is stochastic process. Stochastic process is nothing but it is a type of evolution in which frequencies of genotypes and phenotypes are calculated basing on the mathematical calculations. Here we can take best example like logistic growth curve, of bacteria. It is nothing but calculation of frequencies of genotypes and phenotypes in mathematical calculations. Because if we consider the logistic growth curve, there will be one equation to represent, that is dN by dt is equal to lambda n into k minus n by k. This is one of just the equation. Just try to remember, this was also important equation to study. Another one is determinism. So what is the term determinism is relating to? It is related to the frequencies among variables. Different organisms are there. They are qualitatively and quantitatively different from each other. So frequencies among variations, those frequencies can be calculated. For example, what are the characters that are calculated under determinism? The psychological feelings, the behavior of the organism. We can easily analyze how the person is behaving. how he is expressing his attitude. Such types of terms are expressed in the term determinism. So here the frequencies of variables can be easily quantitatively and qualitatively analyzed. Quantitatively and qualitatively analyzed or calculated. So we can take some of the examples like behavior of the organism, next beliefs of people, etc. Desires of the people, etc. So these are few terms that are mentioned in NCERT textbook. Along with the concepts, these terms are included. So while reading, better to know what are the just meanings of these terms and then read NCERT textbook.